Well, thank you very much. Good morning, everybody. My name's David Baulcombe from Cambridge. Just over 10 years ago, when I was on the Royal Society Council, I was asked to chair a working group to look at science and bioscience and crops and food. And the stimulus for that working group's activity was partly motivated by an awareness that we needed to think about new technologies, and in particular GM, and to assess where science was with that. But it was also stimulated by John Beddington, the government chief science advisor, who came up with the idea of there being a perfect storm associated with food security, and the components of the perfect storm being climate change, population growth, altered demand for food, the sorts of factors that David has just articulated in the previous presentation. And so we set about doing this. We produced a report called Reaping the Benefits, Science and the Sustainable Intensification of Global Agriculture. I think the report was reasonably well received, although the term sustainable intensification, I think, was widely misunderstood. It was a term introduced to the group by one of our working members, Jules Pretty, and we intended it to be applied in a global context. So the awareness was that there was very little, if any, additional land available for food production, but the demand for food was going to increase. So on a global scale, we need to increase the intensity of our food production. There may be some systems where we decrease the intensity, other systems where we increase the intensity, but it was intended to apply in a global context. And of course, sustainability is absolutely key. It's no good producing a lot of food now if you can't do it in the future. Jules came up with a definition of sustainability, which we used in our working group activity: persistence, resilience, autarky, and benevolence. And I hope that these will be fairly controversial definitions of the concept of sustainability. I realize that probably everybody in the room has got a slightly different definition of what they mean by sustainability, but I hope that these general terms are considered to be useful, and they were certainly the guiding principles behind our assessment of the whole situation. The recommendations we made in 2009 were generally under these various headings, and some of them I don't think will be of particular interest to this group necessarily. So there were recommendations to government about funding interdisciplinarity, recommendations about research and training capacity, concern about the difficulty of translating good science and useful science into something that makes a difference in the food production chain. There were recommendations about governance of research in Europe and internationally, and stressing the importance of integration of activities in different regions and countries. We also had a number of recommendations about research priorities, and that's what I would like to consider this morning, to review the research priorities that we identified in 2009, and to ask the question, if we were redoing the report now, what would we change about our assessment of the research priorities? And I just have to preface what I'm saying here by pointing out that I'm today speaking as an individual, not on behalf of the working group, and it may well be that other members of the working group would disagree with me over my assessment of where we stand now. So the recommendations we made about research priorities come under these headings. So one about public sector crop breeding and genomics programs. We saw huge potential in this area of activity, basically breeding and genetic technologies. We identified the power of conventional breeding. We also saw that there were opportunities from GM approaches to increase yield, resistance to stress and diseases. We made a recommendation as well about increased support for ecosystem-based approaches, agronomy, and the related sciences. What we could see was that in science budgets there had been, let's say, good resources of genetics-based science, but these other important areas of science hadn't received the sort of support that we thought as a working group that they needed to have. So we recommended increased support for these ecosystem-based approaches, agronomy, and related sciences. We also identified some long-term high-risk approaches, high-risk, high-return approaches in the genetic improvement of crops, and we thought that those should be well and truly on the table and pursued as aggressively as possible. So what I want to do now is to review these various research priorities that we identified in 2009 and see where we stand now. So the breeding genomics GM-based priorities, these still apply, and they align very nicely with the recent National Academy report, Science Breakthroughs to Advance Food and Agricultural Research by 2030. And looking at this graph from the report, the National Academy report, looking at the impact of various stresses, you can see drought, flood, heat, cold are amongst some of the most prevalent stresses. So in the context of this session related to climate change, we can see that the impact of climate change on crops is huge, and the potential, we believe the potential of breeding genomics and so on and so forth is very great to address those concerns. This was how we saw it in 2009. I think we would reinforce this enthusiasm for these approaches in 2020 for two reasons. So one component that we didn't identify in 2009 was the introduction of speed breeding technologies. And I think we underestimated the power of genomics and the way that genomics would impact breeding. And there are some very nice advances. So speed breeding, a relatively simple innovation that reduces the cycle in breeding programs. Genomic selection backed up by various innovations in sequencing and analysis of sequence data provide huge opportunities, I think, to develop crop varieties that are adapted to the stresses and strains associated with climate change. And I put a couple of references at the bottom of my slide there of what I thought are impressive papers illustrating these points. The other technology that we were not referring to in 2009 because it had barely been developed at that point is gene or genome editing. And I think that when you look at the potential of genetic technologies in 2020, genome editing has to be right up there amongst the advances that you would expect will have huge impact on the technologies. And the group of Dan Voitas have come up with some very impressive advances recently whereby they can bypass the need for tissue culture to develop genome-edited plants. You can get genome-edited plants that are essentially transgene-free in one step. And so if you have advances in crops that are dependent on gene deletions, then the gene or genome editing technologies are basically ready to go. If you want to develop small gene edits where you just change single bases, I think the potential there is also ready to go, applied on a gene-by-gene context, so one gene at a time. targeted replacement, I think, is still a challenge in this technology. I think we need some technical innovation to allow us to, as a routine tool, to replace the sequence of one gene with the sequence of another gene. So that is something that we would be, I think, recommending that needs to be looked at. I think scalability is an important consideration as well. So as gene editing exists at the moment, yes, we can do it on individual genes, but I think that crops don't work like that. The opportunities to make significant improvement to crops just based on individual genes is going to be relatively limited. I think just as with plant breeding brings about innovations in crops because you've changed several genes at once. I think what we would like to have is the opportunity to scale up the gene editing technology so that we can change several genes at one time. But we have to remember that this is a relatively intensive activity, and so whatever gene editing you do, you're likely to apply it to one or two varieties. And one of the concerns that we identified in the 2009 report was the trend towards the lack of diversification in agricultural systems. I think what we would like to do is to somehow get round breeding bottlenecks and to have technologies that allow us to diversify rather than to homogenize crop productions. So still support and still huge potential from genetic and genomic technologies, but some technical barriers to be overcome. One of the other recommendations in our 2009 report was support for ecosystem-based approaches, so crop and soil management and so on and so forth. And one of the examples that we used to illustrate the potential of this approach was one developed by John Pickett at Rothamsted, the push-pull approach, which is basically a companion cropping approach applied to maize in Africa. And it has lots of benefits. It's a really lovely example of integrated pest or crop management because it provides control of the stem borer moth. It provides control of striga, a parasitic weed. It fertilizes the crop. It provides soil stabilization, moisture retention, and so on and so forth. And I think that if we were looking at the situation in 2020 and looking forwards, I think we would be disappointed in the extent to which these types of approaches have been developed and given a science underpinning over the last 10 years. I want to give you one other example here. This is a friend and former colleague of mine, Martin Wolf, sadly died recently with his experimental farm in Suffolk in the United Kingdom, illustrating his experiments with mixtures of wheat. So in these plots, you've got some squares that have mixtures of varieties and some squares that are single varieties. You'll notice the difference in vigor, the mixtures performing much better than the single varieties. And the reason for this is because the blackgrass, a serious weed problem for wheat growers in the UK, is prevalent in the single variety plots, but not in the mixture plots. And I think what this illustrates is that there's real scope for thinking about the sorts of things that we recommended in the report, the ecological-based approaches. And of course, when you ask the question, why is it that the mixtures perform better than the single varieties, the conclusion from that is that there must be stuff going on underground that we don't know about. And that then segues to another area of the report that I think we would want to revise in 2020 compared to the 2009 report, which was, and I actually did a Google search through the report, and we made no reference in 2009 to microbiomes. And that I guess, was not an omission because the science wasn't there now. But now we know a lot more about microbiomes, and we know a lot more about what we would like to know about microbiomes. So we can be descriptive of the soil microbiome, but we can't yet get to an understanding of what is happening in the interactions between the soil microbes and the crops that grow in the soil. There's effects, as illustrated in this slide here, on water retention, on carbon content of the soil, carbon sequestration, and the signaling between the bugs and the plants that influence the way the plants grow. So some of that signaling is nutrient-based, but some of it is hormone and other types of molecules based. And I think, so this is illustrative of how there's a huge gap, I think, in our understanding. And were we writing a report today, we'd want to see a lot more work on soil microbiomes. And I think as well, what we would want to do is to have that work on microbiomes integrated with the work on genetics and genomics. So this is from a paper by the Jeff Dangle group, just illustrating how there is an interaction between the plant genotype on the soil microbiome. And what we would like to do, or what I would like to see happening, is an integration of genomics analysis of the plants with the content of the microbiome, so that we could be more, let's say, scientific and less empirical about our approaches towards exploiting the microbiome. So I think that what I would like to see is integrating genomic approaches with agronomy and agroecology. So developing crops and companion plants that are adapted to intercropping-based systems, and also developing crops that are adapted to nurture the microbiome. So there's a lot of talk about using synthetic bacterial communities as inocular to put into soil. I'm not enough of an expert in this field to be able to comment on the feasibility of that, but I think it's going to be inherently difficult to think about changing the microbiome of a field by spraying stuff on it. But you probably could change the microbiome of a field by managing the crop differently, and also by selecting varieties of the crop that nurture the microbiome. And I think there's huge potential in that area of science. The final point I want to make that featured in our previous recommendations is about grand challenges. So long-term, high-risk approaches to provide high-return targets in the genetic improvement of crops. So we referred in the report to nitrogen-fixing crops, and I dare say Giles will be talking about that a little later on in this session. But I think that were we writing the report now, our enthusiasm for these grand challenges would be reinforced. I think a particularly nice illustration of that is with the work that's been going on in a number of labs looking at enhancing the photosynthetic efficiency of crops. So work from the Hibbard, Steve Long, and other groups. This is an example from the Steve Long group work looking at reducing the non-photochemical quenching in a crop, using a transgenic approach to doing that, and thereby eliminating the non-productive periods of crops in the field. And it seems, at least from experimental plots, that the potential for using to do something that we have thought for a generation or more might be possible, it now looks as if it is possible to develop crops with a much higher photosynthetic efficiency than we had thought of in the past. So I think that this is an illustration of how we should not be shying away from taking on board grand challenges that could be completely disruptive in agriculture of the future. So just to summarize, I think what I'm saying here is that there are many opportunities from genome technology as we recommended in 2009, especially if these opportunities from genome-based technology, and this includes GM and gene editing, are integrated with thinking about crop management and agroecology. And I think that, one thing we should be bearing in mind is really, and I guess this is echoing a point made by David, is that business as usual is not going to work. We are damaging agricultural ecosystems. We've probably all seen the graph of the state of earth players with the High Plains aquifer and seeing the degeneration of that important resource for agriculture. You could look at similar graphs with soil and so on and so forth. If we continue doing agriculture as we do at the moment, the challenge that David articulated in his talk is going to be even more difficult to meet than it need be, because we have the science that can help us change to sustainable practice that will allow us to meet this challenge. So that's what I have to say. Thank you. Yeah.
David, thank you. That was really interesting. Sorry. Okay, is that it? Yeah, good. That was really interesting. Thank you very much. I agree completely with your final statement there about business as usual isn't going to work. It's not going to meet the challenges that David set out in his at the beginning. However, I have to reflect on the fact that you're talking about a 2009 report,
yep,
which probably originated about 2007. So we're about 13 years on, and you have to say, well, how much difference has it made? Has it really hit the ground and made a big difference to productivity in agriculture? And what you're suggesting is, if you don't mind me saying, a bit more business as usual. Okay, maybe a bit of acceleration, but it's not going to get us to where we need to go, because we need to go much, much quicker. Many times quicker than we have over the last decade. So where does the problem sit? Does it really sit in the imagination of researchers, or does it sit in the policy and application side of things and the industry? And I say the industry, and that's a global industry, obviously.
Yep.
And if it does sit in the latter, what the heck can we do about it?
Well, I take your point, but I don't think I am saying business as usual at the research end. I think you're absolutely right to identify the lack of return that society as a whole has had from the huge investment that globally it has put into developing the sorts of science that I've talked about until now. So this tremendous science has not changed the world in a way that many of us practitioners of that science would like it to have done. But I have to remind you of Amara's law, or sometimes it's called Gates' law, which is the observation that generally we tend to over-anticipate what we can achieve in the short term and to underestimate what we can achieve in the longer term. I think we've been guilty in the past of over-anticipating what we can see in the short term. But I think looking forwards, we can see a return beginning to appear on the initial investment. And so I think what I'm saying is we can see that return if we actually move away from business as usual in the research area. So the idea that you get genetics and genomics and link it with agroecology and agronomy and the other aspects of crop production, I think is something that hasn't happened very much until now. And so one of the changes that I would like to see is a shift towards diversifying agricultural systems and using the genetics and the genomics, the opportunities that we have to help us do that. And I could be mentioning data science and all of those things as well. So I take your point absolutely. Business as usual in research will not deliver. We do need to change our research perspective so that we are looking at diversification within production systems.